A very good morning to all of you. Today we will discuss one of the most important topics in microbiology, that is antimicrobial resistance. If we go by the definition of antimicrobial resistance means an ability of a microorganism to stop an antimicrobial working against it. It means ये जो माइक्रो ऑर्गेनिजम्स होते हैं ये एक एंटीबायोटिक को इनइफेक्टिव कैसे बनाते हैं वो हम मैकेनिज्म्स आज देखेंगे दिस एंटीमाइक्रोबियल रेजिस्टेंस में. वन ऑफ द मोस्ट इंपॉर्टेंट मैकेनिज्म्स जो है दैट मीन्स इट ऑकर्स बाय म्यूटेशंस. दिस रेजिस्टेंस मेनली हैपन्स बाय द म्यूटेशंस. एक जनरल म्यूटेशंस आ जाती है या रैंडम म्यूटेशंस आ जाती है माइक्रोबियल डीएनए में, उसकी वजह से ये जो डीएनए जो अल्टर हो जाता है उसका स्ट्रक्चर, तो उसकी वजह से डिफरेंट टाइप्स ऑफ द म्यूटेशंस आकर हो जाती है और उन्हीं म्यूटेशंस की वजह से ये अपने टारगेट साइट्स को चेंज करते हैं माइक्रो ऑर्गेनिजम्स, जिसकी वजह से ये एंटीबायोटिक्स को अपने अगेंस्ट इनइफेक्टिव बनाते हैं. क्लियर. फॉर एग्जाम्पल इफ यू कंसिडर दिस ग्रुप ऑफ माइक्रो ऑर्गेनिजम्स, क्लियर, and if you consider these two microorganisms or these two bacteria are the resistant, and when you give the antibiotic treatment these susceptible bacteria will die and those two resistant bacteria will survive. Clear. What they cause? These resistant bacteria will multiply and give rise to many other resistant bacteria. These resistant bacteria will spread in their community. Clear. Sometimes these resistant bacteria transfers their resistant genes. Sometimes these resistant bacteria transfers their resistant genes to other susceptible bacteria by mainly through the horizontal transfer. There are different processes in horizontal gene transfer, mainly conjugation, transduction, or it can be transformation. Clear. The different mechanisms by which these microorganisms develop resistance, one of the most important mechanisms is the alteration or modification of target sites. Different antibiotics work by binding to their cell wall and inhibit the cell wall synthesis. Some antibiotics work they bind to the ribosomal and inhibit the protein synthesis. Some antibiotics act on the cytoplasmic membrane, and some antibiotics disrupt their different metabolic functions of a bacteria. Clear. But when an antibiotic binds to their cell wall, there is a binding sites mainly for penicillin class of antibiotics के लिए binding sites होती है एक बैक्टीरिया पे. Those binding sites are known as the penicillin binding proteins. Clear. For example, how these alter these penicillin binding proteins and makes these penicillin class of antibiotics ineffective against them. One such mechanism is seen in the some strains of Staphylococcus aureus. Actually, there was an antibiotic known as the methicillin which was developed in 1960s and it was highly effective against these Staphylococcus aureus infections. Clear. But within 1 year it was found that in 1961 it was found that these some strains of Staphylococcus aureus has developed the resistance against that methicillin, and it was mainly found due to this Staphylococcus aureus has acquired a gene. That gene is known as the MAC-A gene. Clear. And this MAC-A gene, what they do by this MAC-A gene, they changed their target sites. By the acquisition of this MAC-A gene, they changed their target sites. For example, this was mainly the normal target size which was present on the Staphylococcus aureus to which the methicillin was binding, and this was, for example, you can consider this as the methicillin and it was binding to this target site. This normal target site was known as the PBP2, but due to the acquisition of MAC-A gene, this Staphylococcus aureus has changed this target site like this. Clear. And this is the antibiotic, same antibiotic, and this antibiotic does not bind to this next target site. This changed target site is known as PBP2A. Clear. And thus makes the Staphylococcus aureus resistant against the methicillin, and this resistant strains of Staphylococcus are generally known as MRSA, methicillin resistant Staphylococcus aureus. Next mechanism which was mainly found in other organisms like Staphylococcus aureus as well, that is enzyme modification, enzyme inactivation. Some organisms, some bacteria or some microbes have acquired an enzyme. That enzyme is known as the beta lactamase. What is the role of this beta lactamase? When an antibiotic comes in close contact with a microorganism, this beta lactamase binds to beta lactam antibiotics. Beta lactam antibiotics are mainly those antibiotics in their structure there is a ring that is known as the beta lactam ring, and this beta lactamase bind to those beta lactam antibiotics and breaks their beta lactam ring open. It means it breaks their structure and makes that antibiotic ineffective. And one such mechanism, another mechanism was seen in mainly due to enzyme modifications. Enzyme modifications was seen in some other microorganisms, like some organisms has acquired a gene. That gene is generally known as the ARM gene. ARM gene means erythromycin ribosomal methylation. By the acquisition of this gene, what this gene did, they changed the target sites of ribosomes. Clear? Actually, this erythromycin binds to the ribosomes. Erythromycin, that antibiotics binds with ribosomes and inhibit the protein synthesis. But what happens by the acquisition of this ARM gene? The target site which was present on the ribosomes. Clear? This ARM gene adds a methyl group at the target site and changed the target site, and due to which these erythromycins are no longer bind to these target sites and makes them ineffective. And some microorganisms naturally possess some protein complexes or some channels. Those are known as the efflux pumps. These efflux pumps have generally the role, they pump out different substrates including antibiotics out of the cell. That means if an antibiotic enters into the cytoplasm of the cell, these efflux pumps will pump out that antibiotic out of the cell and makes and reduce the drug concentration inside the cell and makes that antibiotic ineffective. And there are these were the some of the mechanisms by which these microorganisms develop resistance against different kinds of the antimicrobials. There are different consequences of these antimicrobials. One of the most important antimicrobial resistance, one of the most important consequences of this is that it will lead to the treatment failure. Most important. And it will also account for an increased mortality, death rate. And one of the most consequences is it will add a burden on the healthcare costs. And consider if all these microorganisms developed resistance against all these antibiotics, then what will be the use of these antibiotics? Clear? It will threaten, this antimicrobial resistance threatens us to go back to the pre-antibiotic era before 1940s. Before 1940s, when these antibiotics were not even discovered. This was all for today. Thank you.